I will be discussing some of the work about the Wildlife Connectivity Project, which is being led by the Squamish Environmental Society, the Howe Sound Biosphere Region Initiative Society, as well as a group of local, regional, and provincial partners. I'll be co-presenting with Andrew today as he helps compile and curate the community science data that was used for our project. I'd like to give a nod to the land acknowledgement that was said at the beginning of the webinar, but would like to add that the Wildlife Connectivity Project team is grateful for a partnership and collaboration with the Squamish Ohomeyo as we work towards common goals to protect vulnerable habitat and wildlife movement. I'll just be presenting on one piece of the Wildlife Connectivity Project that I'm leading, but the overarching goal of this project is to safeguard pathways of ecological connectivity in a changing landscape. Wildlife are facing more challenges finding suitable habitat and in order to survive and reproduce, mostly because of the compounding of impacts of climate change and human development. In order to prevent biodiversity loss and protect ecological connectivity networks, we need to understand first and foremost what is suitable habitat for wildlife. So this brings me to the goal of the work that I do for the Wildlife Connectivity Project, which is to use species distribution models, which produce maps of habitat suitability for a focal species. So we'll be using these models to understand how wildlife are using local habitat parcels in the face of changing environmental conditions. And we're working within the Calcim Howe Sound UNESCO Biosphere region and surrounding areas of the Sea to Sky corridor. In order to run these models, we need data. These data largely come from community science at this point. Right now, we have access to two sources of data. We have the Global Biodiversity Information Facility, known as GBIF, which has many different sources of data come together, but this is where our iNaturalist community science data comes from. Andrew will go into more detail about this in a few slides. And we also have access to a camera trap survey that I conducted in our study area for a previous research project. So this is a list of mammals and herpetofauna that we are interested in running our preliminary models for. We hope to expand this list as we get some more data, but the column to the right of the species names show the number of observations that we have currently from from our community. And this was just our first pass through the GBIF database. And we're hoping to kind of get more as we go and improve these models and with time. But now I'll pass it over to Andrew to describe sort of the process of compiling the community science data for our project. And Andrew, if you just want to let me know when you'd like to advance slides, that'd be great.
Sure. Thanks, Kate. It's been awesome working with you and the Squamish Environmental Society on this project. I sort of like to consider my role in this project as being quite humble, but as I mentioned in my presentation, it's non-trivial. That is the curation of biodiversity data. So I'm just going to go over some of the basic considerations for the curation of those data. You can advance the slide there, Kate. So for this project, of course, data curation began with the aggregation and synthesis of biodiversity data for the study area. And in this case, it was fairly straightforward because we drew our data strictly from the Global Biodiversity Information Facility. No doubt, of course, there's going to be more sources of data out there, but we decided to take this approach on a first pass. So GBIF, as some of you may know, is a great filter on available iNaturalist observations, as well as other data. But in the case of iNat, it is very helpful because it aggregates only the observations deemed research grade on the platform. Granted, as Pamela mentioned, this is an imperfect filter, but when you're dealing with very large data sets like this, it's a sensible approach in the first instance. You can advance the slide there. The next step was taxonomic normalization, which is a sort of a data cleaning step where you normalize species occurrences with respect to the names that are used to index them. You can advance the slide again. So in the case of Ursus americanus, the black bear, GBIF recorded you'll find this black bear occurring under so many different names, sometimes including artifacts that just reflect, taxonomic authorities. In other cases, maybe the recognition of subspecies, et cetera. You can move on. For our purposes, we glossed over any potential controversy that might surround the recognition of subspecies within the area and normalize the data to just the species name Ursus americanus. In the case of this project, the recognition of subspecies probably not relevant, but for other purposes, of course, it may deserve more consideration. So next, we undertook some basic filtration steps to ensure the data could be appropriately analyzed in the study. Next. First, we organized the data in terms of the number of records available for each species. The models being developed by Kate tend to be data hungry and sensitive to the number of observations that they include. So this step gave priority to species that had ample data available, while also highlighting where there were gaps that might be filled for certain important species. And then, so yeah, so the black bear, of course, is an example of a species that had over 1,000 observations. So it's going to feature in this presentation, I believe. Next, the all-important matter of coordinate uncertainty, which is often overlooked, especially by contributors to community science platforms. To use biodiversity data for modeling purposes, you want to make sure that your species observations are recorded with an accuracy that corresponds with the resolution of the predictors that you're using. And often we're dealing with a mismatch that can be frustrating. So we heard from Aaron earlier about the amazing resolution of the imagery produced by Planet. But if you want to use Planet imagery to predict species occurrences, you need to have your biodiversity data mapped at an accuracy that corresponds with that fine spatial resolution. Otherwise, you might need to do something like resampling your predictors to a coarser resolution. So coordinate uncertainty is the range of uncertainty represented as the radius in meters surrounding the point that is mapped as the species occurrence. So here you can see as a histogram showing the range of values in the GBIF data for the mammals that we synthesize. And as you can see, most of the records are mapped with a coordinate uncertainty under 50 meters. So these data points can be used to model using predictors with a resolution of 50 meters or more, basically. And some of that, of course, in that bin of 50 meters might be finer resolution, or sorry, higher accuracy. So this is important to note for anyone contributing to community science platforms like iNaturalist, because it is often overlooked. When you map your observations using tools like iNat, make sure you pay careful attention. You might place a point, or if you use a camera that has GPS embedded in it, it might not automatically adjust the range of coordinate uncertainty. So if you're sharing your observations, maybe for this project, place the point, but also notice there's a radius surrounding that point, and you should adjust it to be as accurate as possible to make your data as useful as possible for projects like this. And finally, I guess it's important to note that biodiversity data curation is an iterative process that should be subject to continual review. So I had, for my project or my presentation earlier, I had to gloss over many aspects of this, but it's important not to make assumption about your data and its sources. And this is important not just for community science data, but also historical data sources and any other source. In my bumblebee research, for example, I had to revisit all of the historical records. I went to the Royal BC Museum and UBC and reviewed each of the specimens deposited there, and there were a lot of errors. So again, it's not just community science data that you need to be wary of. So especially where models are being developed to inform decision-making, you want to make sure that you don't take this for granted. Different applications may entail different considerations in quality control and review. And on that note, I'm just going to pass this back on to Kate, who can share more about the considerations made for data used in this project.
Just give you a 2-minute warning.
Okay. Thanks, Andrew, for that. So now, as he mentioned, the model that we use is quite data hungry. But now that we have our data prepared, we can run what's called an integrated species distribution model, or an ISDM. And it maps habitat suitability for a species of interest based on inputs from several different data sources. So to run this model, we first compile our wildlife occurrence observations from GBIF and my camera trap study. These data describe where in our study area we saw or didn't see a particular species. Next, we plot those data onto a suite of environmental predictor variables. These variables describe the conditions of the environment where we either did or didn't see a particular species. So this gives us a gradient of what's considered suitable versus less suitable habitat for our focal species. And finally, we can take that information and predict out to the rest of the study area. So it creates a habitat suitability map, if you will. So it's showing essentially what is the probability of finding a particular species in a particular area. And so that's sort of our proxy for habitat suitability. So I'll quickly present preliminary results for the black bear to show you how community science can be incorporated into modeling work. So here's the regional habitat suitability map for a black bear. You can see the ecological connectivity network forming if you were to connect the red, orange, and green sections of the map. The regional view is really helpful to get a broad picture of habitat suitability, but perhaps you're working at a more local scale. So let's pretend that you manage land in the greater Squamish area, so where this red box is. If you zoom into that red box, you can actually see the same sort of patterns emerging of ecological connectivity. So with this particular model, you can manage land down to a local scale that makes sense for you while still referencing a regional perspective that matters to wildlife. And so what we hope with this model to be implemented in and amongst our study area is we hope that this can be a guide for linking critical habitat parcels at a local scale and creating these ecological connectivity networks. These maps can also be interpreted at multiple scales. You can have the regional perspective as the broadest scale, but you can zoom into any area of interest that's important to you to help you manage land within your jurisdictions. And finally, and perhaps most importantly, this process is iterative and adaptive. So as we improve our database that we're pulling from, as we're getting updated information, as our landscapes are changing with time, we can rerun these models. And so we can kind of, again, continuously incorporate new community science data and new information and feedback. And so this is where I'd like to leave us today. The model works best when coupled with the confirmation of wildlife presence. So we really do rely on community science here. If you have observations that you'd like to upload to iNaturalist, or if you have knowledge of the areas that we're mapping in, this all helps improve the accuracy of our models. And also, if you're thinking about collecting community science data or posting to iNaturalist and you'd like information about best practices, especially to improve locational accuracy, as Andrew mentioned, please feel welcome to reach out to me. I provided my email here, and being conscious of time, I'll leave it at that. So thank you very much.
Thanks so much, Kate and Andrew. That was a great presentation. It's great to see how it kind of fits into modeling work as well. Lindsay, are there any questions coming through?
We haven't got any questions in the chat at the moment, but I invite anybody to do so. I've got a question myself, if you don't mind answering my question.
Sure, sure.
When I was looking at because this is something that we're particularly interested in our project, and we're hoping to obviously steal all your ideas and apply them to our project. But one of the things I noticed there, in terms of, you sort of made the reference in terms of you could see how the model reflected where the results were. Now, my question with that is the fact that human beings are only able to get to a certain amount of the landscape. So is the model basically showing a bias in that? The fact is that it's sort of showing black bears to be concentrated down in the lowlands, but isn't that the human beings are concentrated down in the lowlands rather than the black bear?
Yeah, that's a great question. Something that we're continuing to work with in these models. With this particular model, the integrated species distribution model, there is, unfortunately, with time constraints, I couldn't get into the details of this model, but there is a nice feature where you can actually account for some of the spatial bias of your sampling. So you can incorporate things that bias the detection probability of a particular species. And it helps to sort of draw away some of that bias. And I think one thing we're always looking for, especially since we're incorporating community science data, is we're always sort of putting out a call to action, like to diversify the areas in which we're sampling. So that is something, that we're hoping to, as we improve in these models and as more people are collecting data in understudied areas in particular, it will improve the accuracy of these models. But what we're seeing is where there is data, we can at least get a good start on what is considered suitable habitat. But also, this model does a pretty good job at drawing away some of that spatial bias of where there is data.
Hafs has asked a great question. How is the District of Squamish planning to use this data?
Yeah, that is a good question. Thanks, Hafsa. Yeah, we're working with them, and I think the goal of our work with the Wildlife Connectivity Project is to deliver our species distribution models, in the maps that we create from these models as a tool for them to kind of take and hit the ground running. So we are actively working on creating like a wildlife action plan together with all these project partners. But how we'll do it exactly is still in the works. So I don't have all the details on that just yet, but we're kind of collaborating and working together to come up with a common strategy to implement. Yeah, that's all I can say for now. It's just sort of still happening.
These things always take time to develop, don't they? So, Aaron's put an interesting question here, I'm not sure if you'll know the answer, but do you have a sense as to the minimum data requirements to make this approach work? E.g., how many camera traps location relative to community science observations, or would this depend on the landscape itself?
Yeah, that's a great question, and something that I don't have like a hard and fast threshold of we need exactly X number of observations to make these models work. What I'm finding is I do know at least the ratio of camera trap to community science observations is not you can actually get a fairly accurate model with few camera trap observations, but the presence absence data form of the camera trap observations really is very powerful in this model. So you don't need too many, or you can have them be concentrated in smaller sections of your study area, as I have in this case, and it's still quite powerful in terms of producing a result. I've seen models that have about like around 50 community science observations to be making decent maps. But again, it all depends on how the data is distributed throughout the study area, what this particular species is, and exactly how large the study area is.
So just thinking about, we're sort of in the last 15 minutes and there's no more questions in the chat. So obviously all of our speakers are still online. So if anybody has a sort of a last question to any of the speakers that we've had today, please feel free to take yourself off mute and ask a question, as in the harder you can make it, the better, as in these presenters need a challenge at the end of the day.
Can you hear me okay? My name's Larry.
Absolutely. Hey, Larry.
I have a general question for these amazing contributors here. Once you've consolidated all of this data and you've determined there are areas of very unique biodiversity or species at risk, how do you then proceed from there to protect those spaces from these community contributors and their dogs? Where I am, I identified a unique lomatium species, and 48 hours later, there was a poacher in there had taken half of the population. So how do you protect these sites from the general population and even from some of these community contributors?
Pamela, are you there? Yeah, you've got your hand up, and you seem you'd be a good person to answer this question.
I was like, that's a really good question. It's Larry, correct? Right now, I can only speak for iNaturalist. eBird, probably less of an issue, but with iNaturalist, because you have such a diversity of organisms that are getting reported, and some of them don't move generally, like plants. Or we have slow-moving organisms. Oregon forest snail is another one as an example. It's an at-risk species, and people don't realize that snails actually may be getting poached and collected for cosmetic purposes because the mucus actually has medicinal value. So yes, you can get a nice exfoliation and a facial with snails on your face if you want to pay for that. I don't know if Oregon forest snails are getting poached for that, but obviously we don't want people going in and tromping around in there or removing them because they're concerned that their properties might actually get affected by critical habitat if there's an at-risk species that's identified there. In regards to the lomatium, that one has cropped up before. It's definitely been an issue here. I don't know, Larry, if you're in the Lower Mainland or not, but Tsawwassen First Nation, many families there with that particular Indigenous community have hereditary sites for lomatium plants for harvesting for the seeds. And there's been issues before where the populations have been publicized and then somebody's gone and harvested all the seeds. I'm not aware of the plants being removed completely, but definitely people just taking the seeds because they do have very well-known medicinal value. So one of the things that you can do for reports or any observations that you're reporting onto iNaturalist is you, the observer, can obscure the location of that. There are certain species at risk that iNaturalist, at least iNaturalist Canada, does automatically obscure. There was a process that went on, I think in consultation with COSEWIC a couple of years ago. They came up with a final list. There was a lot of plants on there like orchids and other really sensitive species. I don't think there was much else in the way of like fauna, but bird nests are another example. We know that there's been issues in the past with at-risk species and their nests with avid photographers going in and disturbing them. So definitely using the ability of those tools to hide information. And then folks from, say, the Royal BC Museum or the BC Conservation Data Center, because they're monitoring these platforms quite a bit. If they're interested in finding out the exact location of that species, they can contact the observer directly for more accurate information. Because you, the observer, always have that. Once you have the observation, it's only the rest of the universe that can't see exactly where it is. So I don't know if that's answered your question, but I think in general, when we're all very keen to share information, like the rare bird alerts are a good example. And it typically says, you can't go here because it's private property, so don't attempt to come on here. The owners don't want you on here. The citizen science platforms that we have also have mechanisms to say, we're not going to tell you exactly where this is. So.
Sounds like the best approach to that. So we're getting towards the end of time, but there is another question here from Ali Blake from the Thompson Nicola Conservation Collaboration. So do any of you have any top tips for the TNCC as to how to plan the wildlife connectivity project for the region? It's relatively new and therefore don't have established citizen science projects. What would you suggest? Any of our speakers can take yourselves off mute and answer. So if you were going to start from scratch, no citizen science, where would you start?
Maybe I can, if no one else has a thought, I can jump in here. Yeah, if there's no or very limited community science projects in the area, I think, at least from my perspective, the greatest or first step would be to really make a bunch of connections. You could reach out to local, high schools or universities in the area to try to establish relationships and see if there's any, school projects that could, get launched out of those spaces. You could connect with other nonprofit organizations or municipalities to see what their interest and capacity is in collecting data. And so I think in combination with that and producing educational materials that show a need for this type of work, that would be where I would start to connect with the community and see if you can get some more data through there.
So Andrew just put into the chat there that a great place to start is iNaturalist or GBIF. So even if you don't have a formal group, there's likely to be people out there already collecting and doing community science already. And also Pamela shared a link to the project that the City of Surrey set up around community science, which would help potentially stimulate ideas about how you could start your own project to collect data to feed into the connectivity project.
I guess the other piece that I would say is community science, just think about the other sources that are out there. So Jason Pither at UBCO recently created a connectivity layer that works for all of Canada. So it's a fairly broad brushstroke across lots of different organisms at a fairly coarse spatial resolution. And so it may not be directly relevant for the types of questions that you want to answer, but it's also another way to think about how they did that work and is there something there that's useful as a first pass. So you might think from that, how do you get to the organisms that you're really interested in and the scales, and think about how that matches. So then how do you use that community science to get that specific organism level information or that specific area in there that you think that model isn't doing well, but it's another option anyway, just to consider an existing model. I would share the link, but I don't have it in front of me. But if you send me an email, I'm happy to share that data set with you if you're interested in looking at that broad connectivity analysis.
I've just dropped it into the chat as well if people want to pick it up. Okay, so if I just pass back to Dion for you to be able